In this video, we're going to be covering excitation-contraction coupling at the top there and crossbridge cycling at the bottom. But we actually have to start off going back a little bit and kind of toggling between photos in order to get a better perspective. So we're going to be going back to this original picture that we drew in the last video. We're going to be adding a little bit to it. So you'll remember that when we drew individual thick filaments down at the bottom of this page, that on either end of the thick filament you had these heads sticking out. So now what I'm doing at a more microscopic level, or I guess at a more pulled back level, I'm going to be adding those heads in. And you'll notice the directionality there where the heads are pointing off in lateral directions away from the center of the sarcomere, away from the center of the H zone. Also, that the heads are only present in places where the thin filaments are. That is why the tails are in the center and the heads are on the ends like this. So that is the completion of the picture of the relaxed sarcomere. But down at the bottom, remember that we drew a contracted sarcomere. What made it contracted was that the thin filaments were actually crossing each other. So the sliding filament theory of contraction, recall, is the idea that thin filaments slide past thick filaments in order to start to meet in the center of the sarcomere. And now we're actually looking at exactly how that is done. What I'm doing in this case is I'm drawing these heads from the thick filaments, from the myosin, sticking out and sort of like rowing a boat using oars. They move the thin filaments along. And so as we drew the actin filament down below the thin filament, you know that there's actin active sites. As long as those actin active sites are exposed, the heads will attach, move the thin filament, detach, reattach, move the thin filament, and detach. So in order for those myosin heads to actually attach, we have to remove the tropomyosin blockade. You'll recall when we drew a really microscopic level of the thin filament that we had three proteins that actually made up a part of it. The bulk of the thin filament was the actin with the active sites, with the tropomyosin blockade in place and the troponin or troponin attached to that. But we're going to go ahead and draw it in the way that it would be if the muscle was contracting, which is a condition where the tropomyosin blockade is actually removed. If you think about it logically, you cannot have myosin heads attaching until the actin active sites are exposed. So you have to remove that tropomyosin blockade. So how does that actually happen? Well, we're going to go ahead and draw our troponin or troponin in, and this time we're going to attach it very specifically. One end of it to the actin and one end of it to the tropomyosin. And it turns out that the presence of calcium, as you can see I'm attaching a little pink dot to each of the troponins, the presence of calcium will cause a signal to be sent to the troponin to swing the tropomyosin blockade out of the way. So again, in the presence of calcium, troponin binds the calcium, and then that causes the tropomyosin blockade to be removed and the actin active sites to be exposed, and that in turn allows the myosin heads to bind, and then that in turn causes a muscle contraction. So the next thing we're going to do is kind of a complicated story where we discuss how the calcium came to be present in the sarcoplasm in the first place. So we go back and we're going to draw it not as detailed this time. We're going to go ahead and draw a synapse. We are assuming that you understand that there was an action potential. This is a motor neuron. We had the opening voltage-gated calcium channels, the influx of calcium, which then in turn caused the vesicles carrying the neurotransmitter acetylcholine to bind to the very end of the axon terminal. And now that acetylcholine has crossed the synaptic cleft and is going to be attaching to ligand-gated channels on the postsynaptic cell, which in this case is the sarcolemma of our muscle fiber. This in turn causes the influx of sodium ions, and that influx of sodium ions causes a depolarization, which I just draw with the letter D there. We're going to go ahead and take that depolarization and we're going to send it along the sarcolemma, which is able to take this charge. It's one of the interesting properties of muscle fibers. The sarcolemma is able to pass a charge along its length. So on the one end, actually on both ends, we're going to go ahead and draw some little channels with wings on them, which you'll recall are voltage-gated channels. And so we'll just draw one off to the left to indicate, it's going to be found on either side. The charge is going to be continuing to propagate down the length of the sarcolemma. We're going to draw the ones here with a little more detail just so that we can label them. So we have this charge coming along, and it's going to cause the opening of voltage-gated sodium channels and voltage-gated potassium channels, very much like an action potential on the length of an axon in a neuron. It's really very, very similar. It has a different name and some slightly different qualities, which we don't get into a lot of detail here. But this is not an action potential. In fact, it's an EJP, an excitatory junction potential. It has a different name when you're talking about muscle fibers. All right, let's go ahead and continue that sarcolemma down, and we're going to dive the sarcolemma into a few different T-tubules. And we'll indicate that as that EJP, as that charge is passed down the length of the sarcolemma, it also will dive down into the T-tubules, the transverse tubules. And we're going to continue that story in a moment, but we do have to draw one more cytoplasmic inclusion here. We have to draw one more organelle, which is the sarcoplasmic reticulum. And you'll notice the sarcoplasmic reticula are found very closely opposed to the T-tubules. And I'm going to go ahead and draw another T-tubule and one more set of sarcoplasmic reticula here, oftentimes just called the SR. The T-tubule with the SRs on either side are called triads. Now, the sarcoplasmic reticulum is a storage organelle, and it stores calcium ions with a Ca2+. And so let's go ahead and draw some of our hot pink dots inside of all these SRs here. And we'll go ahead and label the sarcoplasmic reticulum up above. I tried to label it off to the side and I ran out of room. I'll draw it up above here. The sarcoplasmic reticulum is commonly called the SR. It's very close in character to the smooth endoplasmic reticulum of most of the cells of the body. All right, so let's go ahead and follow that EJP, that excitatory junction potential. The charge is going to continue along the sarcolemma and dive right into that T-tubule. When a T-tubule experiences an EJP, it causes the sarcoplasmic reticulum neighboring it to release its calcium content. Now, in this case, we're drawing it such that the last two T-tubules have not yet experienced a charge change. They have not yet experienced an EJP. So in this case, we're going to draw the several thick filaments and thick filaments, some sarcomeres, that have not yet contracted because they haven't experienced the charge yet. But next to that first T-tubule, where it has in fact allowed for the charge, the EJP, to come down to the T-tubule, the sarcoplasmic reticula have released their calcium into the sarcoplasm, and that is going to interact, remember, with troponin on the thin filament, and the troponin will remove the tropomyosin blockade, allowing for the exposure of the actin active sites. And this, in turn, allows the actin filaments, the thin filaments, to move towards the center of the sarcomere, towards the center of the H zone, and this, in turn, indicates that this is sarcomeres that have contracted. Now, in truth, pretty much all the T-tubules would experience the EJP pretty much simultaneously. I draw it this way so that we can see, how it looks when the T-tubule hasn't experienced an EJP, still relaxed sarcomeres, and then when it has experienced an EJP, then you have your contracted sarcomeres. All right, so what we're drawing here, this entire complicated story, honestly, is called excitation-contraction coupling, or EC coupling. I want to kind of pull apart this word for a moment and show you that the excitation is what we've drawn here with the EJP, and the contraction is the actual contraction of the sarcomere. EC coupling, excitation-contraction coupling. The idea that the contraction of the muscle itself is responding to excitation that originally came from a motor neuron. The next part of the story we have to tell is the idea of how does the movement actually occur. And to do this, we're going to get into another detailed story here called Crossbridge Cycling. Now, in order to discuss crossbridge cycling, first off, we have to look at how ATP actually works. And so I'm going to do a real simplified version of this story. We have a molecule of adenosine, which is adenine along with a sugar. But we're not going to draw it in detail here. We'll just write the word adenosine here because the most important part of this story is the 3 phosphate groups that are attached to it. So adenosine has these 3 negatively charged phosphate groups who, if you want to personify the story, become increasingly unhappy and unstable, being forced to bond to other negative structures. So negatives are attracted to positives. They're not attracted to other negatives, to other negative structures. Therefore, as we take phosphate groups and attach them to each other and continue to force them to bond with each other, the bond becomes increasingly unstable and basically has a great deal of stored energy inside of it. The next thing we're going to draw is the hydrolysis of the structure that we just looked at. Adenosine, in this case, is only going to be bonded to 2 phosphate groups, and the 3rd phosphate group has now released. And in the process of releasing, it is going to release a great deal of energy. It's considered an exergonic reaction. So I'll just put a big old E there to indicate. Now the nomenclature here, we have adenosine triphosphate at the top, 3 phosphate groups, and adenosine diphosphate at the bottom, 2 phosphate groups, plus there is, of course, the molecule of inorganic phosphate as well. That is sort of running around rogue at this point and can go on and do other things within the cell. So ATP, adenosine triphosphate, ADP, adenosine diphosphate. ATP has the unstable bond. ADP is a more stable structure, and it also indicates the release of phosphate there. So when we have the hydrolysis of ATP indicated here, we release that loose phosphate into the sarcoplasm in this case, but we also release energy that then goes on to fuel different transactions within the cell, such as the one we're going to be looking at next. So to denote the apparatus that I was just showing you, either ATP or ADP plus inorganic phosphate, they're going to be shown in the crossbridge cycling diagram we're going to be drawing as this, either ATP as a whole structure, unstable, storing a lot of energy, or as ADP with a phosphate has kind of attached to the bottom of it, which means that some of the energy has been released. ATP is less stable, ADP is more stable, but the energy released from the hydrolysis of the ATP allows for in this case, it's going to be the cocking of the myosin head. All right, so when we take a look at crossbridge cycling, we are going to assume that excitation-contraction coupling has already occurred. I don't have the sophistication to draw all of the things from up above and transpose them to this figure. So I'm going to keep this pretty simple here. We're going to assume that EC coupling has occurred, which means that the actin active sites are exposed, which of course indicates that calcium was present and troponin bound the calcium and the tropomyosin blockade was removed. So we have these actin active sites that are exposed, and therefore we have the ability to take this head from the thick filament and attach it to one of these 8 actins in a row that I'm drawing here. And I'm going to go ahead and draw it in the second one for a particular reason, which we'll discuss down the line. The first stage of crossbridge cycling is called crossbridge attachment. It's a cycle, so you could actually start at any point along here and it would still be a story that makes sense. But crossbridge formation is where we have a head which has attached and made a crossbridge with the actin. I wrote this awfully small, so I'll just indicate that what we're writing here is molecule of ATP with the phosphate that has been hydrolyzed. Therefore, there has been energy released into the system, and the myosin head is currently in its high energy state. The next thing we're going to draw is actually a myosin head in its low energy state. where it is bending forward in this case. And since it has bent forward, it has moved. This is where the movement occurs. This is the stage of the movement. This is the stage that actually contraction occurs. It's moved the entire line of actin down the line. I actually ran out of room to draw them all. This stage is called the power stroke. Now, the power stroke, ironically, is the relaxation of the myosin head. So we'll draw a few arrows indicating the movement, the directionality of movement, both of that myosin head and of the entire actin filament. And we're also going to go ahead and draw our loose ADP and our loose phosphate group that have now popped off. And because they've left, the myosin head is no longer in its high energy configuration and has relaxed forward, if you will. The next stage, we're going to go ahead and draw our myosin head still relaxed forward, but in this case it has attached a molecule of ATP. There's a little binding site on the myosin head that allows us to bind either ATP or ADP, whatever needs to be bound. And so we're going to say, well, the myosin head is still leaning forward. The actin filament has still been moved forward. This is called Crossbridge detachment. And what actually caused the detachment of the myosin head from the actin filament was the binding of a whole molecule of ATP. Now, it hasn't hydrolyzed yet, so it hasn't released its energy. But it turns out just the binding of the ATP to the head is enough of a shape configuration change that the head feels free to release from the actin filament, from the thin filament. And the next stage, what's going to happen is you'll notice that that head is leaning backward again. And so it was leaning forward in the previous picture, and now we're going to draw it leaning backward again. The actin filament, the thin filament, is still shuffled along. It has still moved along from where it was in the power stroke. But now we're going to cock the myosin head backward. Now, how did that happen? It turns out the cocking of the myosin head is its high energy configuration. And the way that we put energy into a system, if you revisit the ATP story, is we go ahead and hydrolyze the ATP to ADP and inorganic phosphate. And this is the cocking of the myosin head. Now, to revisit the Crossbridge cycling, I would suggest that you look through all 4 stages, maybe starting at different stages each time. Remember that it is a cycle, so it makes sense to start at any point along the way. You'll want to engage with the ATP versus ADP story. You'll want to engage with where the myosin heads are in their high versus low configurations. And then you'll want to think about the moving of the actin filament as well. You'll notice that when we had what we called stage 1, the crossbridge formation, we were engaging the myosin head with actin number 2 of our 8. But when we came all the way to what we called stage 4, the cocking of the myosin head, the next time we're going to come around to the cycle and begin it, it's actually going to be engaging with number 7 of 8 of the actin filament. So we've kind of shifted things down a way. So you want to engage with the story in that way as well.